We'll start in just a few minutes. Right, I'll start by sharing the slides. Right. Welcome to the 10th live session of structural biology. In this session, we will be talking all about molecular dynamic simulations. Firstly, molecular dynamics is a process to study and analyze the dynamic nature of a system. The tool to perform molecular dynamics is computer simulation. And this computer simulation is the process of mathematical modeling performed on a computer, which is designed to predict the behavior or the outcome of a real or physical system. So basically, if you have a real system, you can perform different experiments and get experimental results. Based on the real system, you can develop a model and a model system. And on this model system, you can perform different simulations on a computer. This is generally done when performing experiments is either very time consuming or costly. So this model system can give rise to a theoretical hypothesis, which can be tested by these computer simulations. And then you get the results from the simulations and you compare them to the experimental results as well as the results that were predicted from the theory. So using all these three, the experimental results, the simulation results, and the prediction from theory, better models can be made as well as the theoretical assumptions can be improved. So this is an iterative process. Once you have the initial set of results, you can use them to improve the initial model and get better results. And they can also help to screen a large number of, like if you have a large number of possible targets for a drug, screening them using experiments might be very time consuming. So the initial screening can be done using these simulations. And then the experiments can be performed to find out which of them has the best interaction with the drug. So experiments and simulation kind of go hand in hand. So in molecular dynamic simulations, the time-dependent behavior of a molecular system is observed. So in all the experimental techniques that we have seen so far, like X-ray crystallography, NMR, or cryo-electron microscopy, generally you observe the system at a particular time or a snapshot of the system. But molecular dynamic simulation looks at the time-dependent behavior and how the system changes with time. It provides detailed information on the fluctuations and conformational changes of proteins and nucleic acid. So how the protein folds and and how it moves so that it can interact with different ligands is observed using this molecular dynamic simulation. It investigates the structure and dynamics and thermodynamics of biological molecules and their complexes. So we will go into more details of that in the later slides. So protein folding and protein ligand interactions are two important biological problems that were solved using this molecular dynamic simulation. So if you look at this particular protein shown here, molecular dynamic simulation was carried out to find out how this protein folds. Firstly, the thing you have to understand about folding of proteins is that there may be numerous unfolded states, but there is only a single native folded state. So if you look at this energy profile diagram, where the free energy is plotted along the z-axis, and the x and y-axis are the conformational space. So you can see there are 4 unfolded states, and they are all of higher energy. But the global minimum of the system, which is the lowest possible energy, is the one that belongs to the folded native protein. So considering this, the mechanism of protein folding was studied using molecular dynamic simulation for this particular protein. And it was observed initially when the time is 0, these two helices, 1 and 4, align with each other. Then as the time proceeded, the different conformational elements of the final native state came into being. So at 18 microseconds, these two helices shown in blue, 2 and 3, came into alignment. At 18.9 microseconds, a loop formed and eventually, as around 20 microseconds, the final native structure was observed. So molecular dynamic simulation gave a picture of how the protein goes from the unfolded state to the folded native state. Also, molecular dynamic simulation can be used to study protein ligand interaction. So what this first diagram shows is a simulation box containing a mutant of the T4 lysozyme binding to a molecule of benzene. So this has been shown enlarged over here, where you can see this protein, this particular protein is binding to a benzene molecule, which is shown in red over here. And what this diagram shows is that as the concentration of benzene increases, like if you see this bar diagram from blue to red to violet, as the concentration of the benzene increases, the binding constant also increases. So molecular dynamic simulation can even give you an idea about the binding constants and what is the optimum concentration of benzene that has the highest binding constant to this protein. So using molecular dynamic simulations, different ligands were compared, like benzene, indole, toluene, ethyl benzene. Different ligands were compared to see which had the best binding affinity to the protein. So all these kinds of thermodynamic studies can also be done using molecular dynamic simulation. And this is mainly used in designing drugs, because to find out the optimum protein ligand interaction to have the most efficient drug, molecular dynamic simulation can be used to find the most efficient binding constant. So why do we do simulations? In some cases, experiment is impossible. Like if we want to study what is happening inside the stars, or if we want to predict the weather. In these cases, experiment cannot be done. Or maybe the experiment is too dangerous. Like suppose we want to study what happens during a plane crash or during an explosion. Or the experiment is very expensive, like simulating a high pressure region or a wind channel. Or we want to study something that is happening on a very short time scale or a very small area. So experiment cannot look into that very short time scale. So, or that very small area. So in these cases, simulation comes handy, because in simulations, the parameters are in your control. So even if the real activity is happening on a very short time scale, the simulation can slow down the time scale so that you can observe what is happening. Or it can, or if the activity is happening in a subatomic space, the simulation can enlarge that space and magnify it so that you can observe what is happening. So these are the advantages of simulation, and why in many cases, simulation is preferred over experiment. So these are the experiments. We have learned many different kind of experiments like NMR or diffraction. What you need to understand is that experiment and simulation support each other. So the experimental results can improve the initial model of simulations, and the simulation results can help in choosing what molecules to experiment on, which will give the best results. So they go hand in hand. So why do we do molecular dynamic simulation? Molecular dynamic simulation is the most realistic simulation model, and it can provide structural and dynamic information, and we can study both the native as well as non-native conditions. So as we discussed, this experimental data as well as the simulations combined together to give an idea about the thermodynamics and kinetics of the system and how the protein is folding or how it is binding to a ligand. So you can look at this GIF here, where molecular dynamic simulation will basically study how the molecules, so this molecule, as you can see, is coming down and it is interacting with all the other molecules. So it will give a picture of how what is the activity that is going on in the subatomic space and how it is varying with time. So now we will look into the experimental and theoretical states of a protein. So mainly there are three different ways of looking into how a protein is working. One is the in vivo method, in which the protein is inside a living organism like a bacterial cell, and it is studied while it is inside that living environment. So that is called in vivo, and it means that the study is taking place in a living organism. So what happens in vivo is that you can have many different kinds of proteins. So you are not able to isolate that particular protein that you want to study. So that might create some confusion. In vitro, so we had learned about in vitro, where in a plasmid using restriction enzymes, a particular fragment is cut out and replaced with the gene that we want to replicate. And in this recombinant plasmid, it is allowed to multiply, and then the protein is isolated, and by gel chromatography, gel electrophoresis, suppose this is our protein that is shown in red here. So using gel electrophoresis, that particular protein is cut out, and you are isolating only the protein you want to study. So that is in vitro, which basically means the study is taking place in a test tube. So you are able to isolate only the protein you want to study, and that clears up a lot of confusion. The final method is in silico, and it means that it is performed in a virtual setting, that is a computer or virtual simulation. So in silico, so in vitro, you have the protein you want, but you have say thousands of molecules of that protein. But in silico, you can study a single molecule of that protein, because you are studying it in a simulation. So you can isolate out a single molecule, and you can study how that single molecule is folding or interacting with a ligand. So this gives an even more clearer picture of what is happening. In silico is an expression used to mean performed on computer or via computer simulation. In silico studies has been widely used to predict how drugs interact with body and with pathogens. For example, a 2009 study used software emulations to predict how certain drugs already on the market could treat multiple drug-resistant and extensively drug-resistant strains of tuberculosis. So the advantage of in silico is that is mainly in this drug screening. So basically, if you want to experiment the effect of, say, hundreds of drugs and find out which is the most efficient, it will take a long time and be very expensive if you do it in vivo or in vitro. But in silico, you can screen hundreds of drugs in just a few days, because it is the computer that is running the simulation, and it will narrow it down to the most efficient drugs, which can then be tested in vivo or in vitro. So this is the basic process of molecular dynamics simulation, and comparing that to the experimental process. So in the experiment, you prepare the sample, and then you connect the sample to the measuring instrument, like say an NMR spectrometer or a cryo-electron microscope. And you measure a certain property for a set time interval. Like NMR is measuring the chemical shifts, or you can even measure the dynamics using cryo-electron microscopy. And finally, you minimize the noise. So in experiment, there can be a lot of like signal is to noise ratio can be very low. So you have to improve the experiment, so the signal is to noise ratio is optimized. In molecular dynamics simulation, what happens is you have a system of particles, and you have to solve Newton's equations of motions, which I'll discuss in the subsequent slides. And from that, the position and velocity of each particle is extracted, and then statistical mechanics is used to extract different properties, like root mean square deviation during folding of a protein, or the protein dynamics. So all this will be discussed later. the first step in molecular dynamic simulation is to divide the whole time period into discrete time steps. So if you're studying, say, the movement of the system in 1 nanosecond, snapshots of the system are taken every 1 femtosecond. And there are different time scales of this simulation. So if you want to study bond stretching, it falls between the picosecond to femtosecond time scales. The elastic vibration of proteins falls around the nanosecond to microsecond time scale. And you can see for different proteins, the folding time scales are different. Like for general protein, it is around the second to millisecond. But if it is a purely beta hairpin protein, it is microsecond and longer. But alpha helix folding occurs on a shorter time scale. So depending on the time scale of the folding of the proteins, you have to choose a certain time scale to carry out the molecular dynamic simulation so that you can clearly observe the folding process. So a study of, now we'll go into the study of protein folding. Instead of folding of protein, we study unfolding of protein using molecular dynamics. So why is that? Because using already established experimental information in the folded 3D structures provides more authenticity to the simulation experiments. So what does this mean? So if you want to study unfolding of a protein, you have to start from the folded structure. And the folded structure initial model that you will build is based on the experimental structure that is observed from NMR crystallography or cryo-electron microscope. So you have a very good initial model for the folded structure. So it is easy to go from that to the unfolded structure from using computer simulations. However, if you want to start from unfolding, it is very difficult because constructing the initial model of the unfolded structure is very difficult. Because so far, NMR crystallography or cryo-electron microscopy has not been able to give us a very good idea about the unfolded state. So because we need a good initial model, it is always good to start from the folded structure and using simulation study how it is unfolding. Then, of course, we can get the reverse picture also, that how the unfolded structure is going to the folded structure. In molecular dynamics, a molecule is described as a series of charged points, which are the atoms, linked by springs, which are the bonds. So if you picture each of these blue dots as the atoms, these are a series of concentrated charges. And the bonds that are between the atoms are thought of as springs. So the bond length is not considered constant, but like a spring, it can expand or compress. To describe the time evolution of bond lengths, bond angles, and torsions, and the non-bonding van der Waals and electrostatic and hydrogen bonding interactions between atoms, one uses a force field. So this force field is very important during molecular dynamics simulations because the force field will allow you to simulate theoretically all the energies that are present between different atoms. So, different atoms can have bonded interactions like covalent bonds or non-bonded interactions like van der Waals or hydrogen bonds. To study all these interactions, a force field is needed. And a force field is a collection of equations and associated constants designed to reproduce molecular geometry and selected properties of tested structure. So basically, the force field will reproduce the interactions that are present between the different atoms, and that will help to accurately model what is happening in the real protein. So as I said, there are different kinds of interactions. So the first one is called, so first we'll study the different kinds of bonded interactions. So if there is a bond between two atoms, this bond can stretch or compress like a spring. So this is a graph in which in the x-axis you have the distance between the two atoms, and in the y-axis you have the energy of stretching, which represents the energy required to stretch or compress a covalent bond. So there is a certain distance at which the energy is minimum. and that is the equilibrium bond length. So that is the bond length that is most stable and which generally exists in the molecule. If you either go to longer lengths, that is, you are stretching the bond, the energy will increase. Or if you go to shorter lengths, that is, you're compressing the bond, the energy will again increase. So on either side of the equilibrium bond length, the energy will increase and it follows the Hooke's potential for an ideal spring. This energy of stretching is represented by a formula which is half K Rij minus R0 whole square, where K is a constant which depends on the kind of atoms involved and on the type of bond, if it's a single bond or a double bond. And Rij is the position of the bond at any particular point, and R0 is the equilibrium bond length. So as you can see, as the Rij either increases or decreases from the equilibrium bond length, the energy of stretching will increase. The second kind of bonding interactions is called bending. So if you have a bond between three atoms like I, J, and K, there are two bonds between I, J, and JK. There is a certain equilibrium angle between the bonds which is the most stable. So that equilibrium angle is called theta 0. Now, energy of bending is the energy required to bend the bond from its equilibrium angle. So from the equilibrium angle, if you go to larger angles, the energy increases, or if you go to smaller angles, the energy also increases. This is also based on the potential energy of a spring, and it has a very similar formula as the energy of stretching. Only difference is that in stretching, it is the bond length, and in bending, it is the angle between two bonds. So this energy of bending depends on the chemical type of the atoms. The third kind of bonded interaction is a torsion. So distortion of the torsion angle. So we have learned that a torsion angle is formed when there are four atoms. Like suppose A, one second. So suppose this is your A, this is B, this is C, and this is D. So the angle between these four atoms form your torsion angle. So basically it is talking about the rotation along this bond, the middle bond. So torsion angle is defined by the angle of rotation along the middle bond, which connects. So there are three bonds connecting four atoms, and the angle of rotation along the middle bond is called the torsional angle. That is also defined by a formula which has a constant K and 1 minus cos n phi, where phi is the torsional angle. So the energy of bonded interactions is calculated by adding up the energy of the stretching interaction, the energy of the angle bending interaction, and the energy of rotation along bond or the distortion of the torsional angle. So there are three kinds of bonding interactions. Now we'll go to the non-bonded interactions. The first of that is the van der Waals interactions. So suppose there are two atoms. When they are very far away from each other, the interatomic energy is 0. As you come closer, the distance between the two atoms decreases. The energy becomes more negative. So there is an attractive energy. Sorry, I think someone wants to join. I'll just allow them. Right, I'll continue. Right. So as I was talking, there are different kinds of non-bonded interactions. The first of which is the van der Waals interaction. So when two atoms are at infinite distance from each other, there is no energy between them. But as they come closer, a certain attractive energy starts working between them, which reaches a maximum at a certain distance. and that is the equilibrium van der Waals distance between the two atoms. If you try to bring the atoms closer than that, the repulsive energy comes into action and the energy increases tending to infinity. So this is a van der Waals interaction between atoms, and the frequently used formula for that is basically a constant divided by r to the power 12 minus b by r to the power 6. A and B are constants, and R is the van der Waals distance between the two atoms. So as you can see, the repulsive energy increases much more sharply than the attractive energy. There are other kinds of van der Waals interaction, like the Coulombic interaction, which is basically the electrostatic interaction, and it depends on Q1, the two charges between the atoms. So if there are two atoms, I and J, Qi and Qj is the charge of each atom, and the energy is directly proportional to these charges. And it is inversely proportional to the distance between the atoms. So if you have two oppositely charged atoms, as the distance between the atoms decreases, the energy becomes more and more negative, because the product of Qi and Qj would be negative, giving an attractive energy. That is, the net energy is negative. Again, if the two atoms are of the same kind, like two hydrogens, the two charges will be of the same sign. And so the net sign of the energy will be positive. And so as the distance between the two atoms decreases, the energy will become more and more repulsive. That is, it will become more positive. The other kind of non-bonded interaction is hydrogen bond. And you can, in proteins, hydrogen bond can occur between the acidic amino acids and the backbone amide or the basic amino acids. So it is primarily the electrostatic force between the hydrogen atom and an electronegative atom like oxygen or nitrogen. And the formula for the hydrogen bonds is again based on the interatomic distances of the hydrogen and the electronegative atom. So it is C by R to the power 12 minus D by R to the power 10, where R is the distance between the two atoms. So the total energy is basically summing up. So we talked about the bonding interactions. So you have to add up the three kinds of bonding interactions, which is the stretching, the angle bending, and the torsional distortion, or the rotation along the bond. And the three kinds of non-bonded interaction, which is basically the van der Waals interactions, the electrostatic interactions, and the hydrogen bonds. So once these six kind of interactions are added up, that gives you the total energy of the system. So what are the major steps of this molecular dynamics simulation process? So the goal is to develop a model system from our understanding of the real system. So you get an idea of the real system from experiment, which is NMR cryo-electron microscopy or X-ray crystallography. You get an idea of the real system, and based on that idea, an initial model is developed for the computer simulations. Then the computer simulations is done, and the experimental and simulation results are compared. And using this comparison of the experimental and simulation results, a better initial model is designed. For example, if the experimental and simulation results are varying, that means the initial model of the simulation has to be improved. And like that, the experiment can help in getting a better initial model. And the advantage of simulation is that there are many cases where the experiments are problematic. So like I discussed, there are many cases where the say protein folding is occurring on a very short time scale that can't be observed experimentally. So those are cases where the simulation comes in handy. So the basic steps of simulation. So the first step is to build a realistic atomistic model of the system. So in this first step, there are several sub-steps. The first step, sub-step, is to get a valid protein structure. So this can be gotten from the different experimental studies like protein crystallography, NMR, or cryo-electron microscopy, or even from theoretical studies like the modeling studies. So using these, a valid initial protein structure is built. then the structure has to be repaired. So in many cases, residues or atoms may be missing. Like suppose this is a crystal structure that was obtained, but the 17th residue was missing. So between cysteine 16 and valine 18, the residue was missing. So using Wynkoop, which we discussed in the previous class, so this structure can be refined to find out which residue can fit in this space. And it was found that methionine is fitting the best at the 17th vacant space. And so the structure was improved by identifying and inserting this missing residue. Secondly, if there are incorrectly modeled residues or groups, that can be corrected. So from the electron density obtained from diffraction studies, it was observed that this initial structure that was predicted was wrong because it is not fitting with this electron density. But then the structure was corrected and it was observed that if a phenyl ring is present, that is this structure is present, then it fits much better with the predicted electron density map. So like this, many different wrongly modeled residues or groups can be corrected. Then the protonation states need to be homogenized such that if we are studying the system in a particular pH, the protonation states of the acidic or basic residues must correspond to that particular pH. And the different rotamer conformations and steric clashes are also corrected. And the third substep in preparing this initial model is to delete hydrogen wherever required. So since there are so many hydrogens in a protein structure, often it can confuse the results or give us unwanted artifacts. So the hydrogens are deleted wherever we want a more clear picture. And we can delete the solvent if we want to study the folding of the protein in the absence of any solvent, and we can clean up the structure in that way. So now that we have followed the three sub-steps of getting the initial protein structure and then repairing and cleaning the structure, now we have a clean PDB structure. So the second step is to simulate the behavior of the system over time using specific conditions like temperature, pressure, and volume. So this is based on Newton's equations of motions. So as we know that the force applied on an atom is the product of its mass into the acceleration. And acceleration can be written as the double derivative of its position with time. So this acceleration can be replaced by the double derivative of the position r at time t. So using this, we can find the force on an atom. The force can also be represented as the gradient of the potential energy. So the change in potential energy with time, taking the negative of that, is also another expression of the force. So now we can equate equations 2 and 3 and get this equation 4, where the gradient of the potential energy with time is equal to the mass into the double derivative of the position at time. So now that we have the force acting on each of the atoms at a particular time, we can get the trajectory of the atoms. So the most common way to get that is called the Verlet algorithm. This algorithm uses the positions and accelerations at time t and the position from the previous step to calculate the new positions. So at time, suppose we know the position at time t, then this is the equation that is followed, where the position at time t plus delta t will be twice into the position at time t minus the position at time t minus delta t plus delta t square into the acceleration at time t. So using this formula, the position at different times which have a time gap of delta t can be observed. And if we connect all the positions, then we'll get a trajectory of the atom. So how is this delta t selected? The general rule is that the time step is approximately 1 order of magnitude smaller than the fastest motion. So if we want to study hydrogen vibration, which has a time step of 10 femtoseconds, then the delta t chosen has to be 1 order of magnitude smaller, which means it has to be 1 femtosecond. So choosing the appropriate delta t, we can calculate the position at different times separated by delta t. And then connecting all the positions, we'll get an idea of the trajectory of the system. But before all this, you have to make the PDB file consistent with the simulation. So we have the PDB file. What we need to prepare are two kinds of files called the topology file and the parameter file. So basically, why we need this? Consider the diatomic hydrogen molecule. In molecular dynamics simulation, this molecule is modeled as 2 point masses connected by a spring. So two sets of information are needed before we can simulate the hydrogen molecule. Firstly, we need to define which atoms are connected through chemical bonds. So that information is provided by the topology file. So topology file basically specifies the different bonds. So if there are two atoms connected by a bond, the topology file will specify that. It also specifies the angles, which are basically three atoms connected by two bonds. What is the angle between the two bonds? And it will specify the dihedrals. So if there are four atoms connected by three bonds, what is the dihedral angle, meaning the angle of rotation along the middle bond. But actually, all three of these don't need to be specified separately, because if we have a complete list of two atom bonds, then automatically from that, the angle and dihedral information can be obtained. Because if you know all the two atom bonds, then automatically you will know these two bonds, which will give you the angle, and you will know these three bonds, which will give you the dihedral angle. However, if there are certain improper angles which are used to restrain chiral and planar systems, that must be specified. So suppose this is a pyramidal system. So this is a special case, and in this case, the angle needs to be specified, because suppose we want to find the angle between these two atoms which are not directly connected by a bond. So in that case, the angle alpha will need to be specified. The parameter, so the topology file is giving all the information about which are the atoms connected by bonds. The parameter file will quantify the variables which are used in the force field potential energy. So we learned about the different kinds of bonds between atoms, both bonded and non-bonded. And we had seen there are different kinds of constants, like the stretching constant, the angle bending constant, or the Coulombic constant. So the parameter file will give the values of all these constants, which represent the stiffness and the equilibrium value of the angle. So the equilibrium bond distance R0 or the equilibrium angle theta 0, and as well as all the constants which specify the stiffness of the bond will be specified in the parameter file. So these two files need to be created before we can simulate a PDB file. So if we want to find the parameter file of a non-standard biological molecule, so it is often possible to run simulations using existing parameters for the standard amino acids. So in all the molecular dynamics simulation software available, you will find existing parameter files for standard amino acids like lysine. But suppose your protein has an ornithine, which is basically lysine, but with one CH2 less. So lysine has 4 CH2, then NH2, and ornithine has 3 CH2 and then NH2. So you can modify the parameter file, because the coordinates of the first 3 CH2 will be the same, and the coordinates of the NH2 can be predicted from lysine. So you can modify the parameters of lysine itself, and you don't have to put time and effort into developing a new parameter file. Next, we have to look at the solvation models. So why is solvation required? This is because many biological processes occur in aqueous solution. So most of the proteins, they are folding in the cellular environment, which is a mostly aqueous environment. So it is not like it is folding in vacuum. So in order to simulate what is happening in reality, we need to create an appropriate solvent condition, because solvation plays a crucial role in the molecular conformation, electronic properties, and binding energies. So there are two kinds of solvation models. The explicit solvent models relies on using hundreds or thousands of discrete solvent molecules. Such calculations converge only slowly because of the large number of particles involved. So in explicit solvent molecules, in the simulation model, you have to place hundreds of thousands of different solvent molecules. And because there are so many solvent molecules, the simulation will take a large time to run, but it will be more accurate because it is more close to the real system. Implicit solvent molecules treat the solvent as a continuous medium having the average properties of the real solvent and surrounding the solvent beginning at the van der Waals surface area. So if you see this outline, this is the van der Waals surface of the protein, and away from that outline, a continuous medium is taken, which is defined as the implicit solvent. So the advantage of this is that this simulation will take less time since you have taken it as a continuous medium and not as separate molecules. But the disadvantage is that it is less accurate because this is an approximation of what is the real, what is existing in reality. Because in reality, solvent is many different molecules and not a continuum. The next thing that is needed is to define a periodic boundary condition, or PBC. So PBCs are a set of boundary conditions which are often chosen for approximating a large system by using a small part called a unit cell. So like we had studied in crystallography, we can understand the properties of the crystal by studying only a unit cell. So similarly, just 1 second, I think my connection is breaking. Yeah, okay. Yeah, it's fine. Yes, so like I was saying, in crystallography, we had studied that we can study the properties of the crystal by studying only the unit cell. So similarly, if you have many symmetric arrangement of molecules, you can choose a particular area which represents the rest of the entire system. So suppose you have a molecule moving sideways and a molecule moving down, and this is the condition that is there in the entire system. So if you study the dynamics in this particular cell that is in the center of the system, that is enough to understand the dynamics of the entire system. PBCs are mostly used in performing simulations and mathematical models. In molecular dynamic simulation, PBC is applied to calculate properties of bulk liquid crystals or mixtures. So only when you have a homogeneous system, like a liquid crystal, can you use this condition of PBC. Because if it's a heterogeneous system, you cannot, there won't be a single unit cell which is representative of the properties of the entire system. And PBC is generally used to simulate solvated macromolecules in a bath of explicit solvent. So from PBC, there comes the concept of central simulation box. The central simulation box is extended to infinite dimensions by considering its periodic images. For instance, this blue particle here has neighbors within a radius shown by this light orange circle. And these neighbors are periodic images of particles from the central box. So this central box is representative of the entire system. And if you have the picture of the molecular dynamics of the central box, you can translate it into various directions and get an idea of the entire system. Next, we will talk about the concept of phase space and ensembles. So, what is phase space? In dynamical system theory, a phase space is a space in which all possible states of a system are represented, with each possible state corresponding to one unique point in the phase space. For a model system, the phase space usually consists of all possible values of position and momentum variables. So we saw in the previous slides that using Newton's equations of motion, you will be able to find the position and momentum of a particle at a certain time. So all these position and momentum variables of all the particles constitute the phase space. A given atom is represented by its time-dependent position and momentum vectors. For n atoms at a given instant, if you consider a plot of all the positions and all the momentums, then that will be the phase space. So phase space is basically a space which gives you an idea of all the position and momentum variables of all the atoms at a particular time in the system. An ensemble is a collection of all possible systems which have different microscopic states, but an identical macroscopic or thermodynamic state. So in the ensemble, you can have many. So suppose an ensemble is a homogeneous solution. So in that homogeneous solution, you have many microscopic states, which are each separate atom is a different microscopic state. But the homogeneous solution has the same macroscopic state because the solution will only have a single temperature, a single volume, and a single pressure. So the macroscopic properties of the solution are all same. There are different kind of ensembles that we will come across, like the microcanonical ensemble, which has a constant number of atoms, volume, and energy. So in the microcanonical ensemble, it is sealed. So the number of atoms cannot escape and it is constant. Because it is sealed, the volume is also constant and there is an insulation at the sides of the container. So the energy is also constant because the energy cannot escape. In the canonical ensemble, it is similar to the microcanonical, but the insulation is not present. So because the insulation is not present, the energy will exchange with the surroundings and the temperature will equilibrate till the temperature is the same as the surroundings. So in the canonical ensemble, energy is not constant because the insulation is absent, but the temperature is constant because it can exchange energy with the surroundings and maintain a constant temperature. In the grand canonical, and there are other kinds of ensembles which are not that important right now, but all of them have different criteria of parameters that are maintained at constant. Next, we will talk about energy minimization. So in the field of computational simulation, energy minimization or geometry optimization is the process of finding an arrangement in space of a collection of atoms where, according to some computational model, the net interatomic force on each atom is acceptably close to zero, and the position on the potential energy surface is a stationary point. What this means is that it is the most stable state of that system, where it is the lowest energy state or the most stable state of that particular system, and which is the result of the energy minimization. This is based on a theory called Anfinson's theory, which states that the protein native structure corresponds to the state with the lowest free energy of the protein solvent system. So Anfinson had done an experiment. So what he had done is he had taken a native ribonuclease, which had different disulfide bonds. So if you look at these bonds here, these are the disulfide bonds, 4 of which are shown in this diagram. Disulfide bonds are formed between 2 cysteine residues. So he had taken a reducing agent, or you can heat the system or add urea. So all either adding reducing agent or heating or adding urea, what it does is that it breaks the disulfide bonds and the protein unfolds. After that, he had cooled the system or removed the urea to allow the protein to fold back, and it had folded back into the same native conformation, and the four disulfide bonds were in the exact same locations as it was there in the original structure. So from this experiment, he had concluded that the protein native structure is always the lowest energy state of the protein, which is why when an unfolded protein is allowed to fold back, it will always fold back into that particular native structure, because all systems always tend to go to the lowest energy. Now, a potential energy surface is a plot of the mathematical relationship between molecular structure and its energy. The surface might define the energy as a function of one or more coordinates. So like you can see, this is a potential energy surface that is shown, where potential energy is in the y-axis, and these are called variation of parameters. So this can be the position of the, different parameters vary while when the protein is going from the unfolded state to the folded state, and that is represented in the x-axis. So an unfolded state is generally very unstable. So if that is the starting point conformation, and a simulation energy minimization is carried out, then it can encounter local minimum. But a good energy minimization simulation will not stop at local minima. It will continue the simulation till it reaches the global minimum, which according to Anfinsen is the native structure of the protein. So in this energy minimization, the protein, what the simulation will do is that it will continue changing the parameters till it reaches the global minimum. And that is how it is able to predict the native state of the protein. This potential energy surface is a conceptual tool for aiding the analysis of molecular geometry and chemical reaction dynamics. One of the necessary points are evaluated on a potential energy surface. That is, the points can be classified according to first and second derivative of energy with respect to position. So I'll talk about that in the next slide. So basically, if this is your potential energy surface, you can have a maxima, which is represented by this red point. You can have local or global minimum shown by this green point. Local minimum is this blue point and global minima is this green point. Or you can have different you can have a transition state. So how these are distinguished? Basically, if you have the energy profile is like this. This is a minimum. Whether it is global or local, we'll come to later, but this is a minimum energy. So the characteristic of that is that the double derivative with respect to x or y are both positive. So basically, if you want to find out if a particular point is a minimum, the double derivative of the energy at that point with respect to x coordinate and y coordinate has to be taken. And if both are greater than 0 or positive, then it is a minimum. If both are less than 0 or negative, then it is a maximum. And if one is greater than 0 and one is less than 0, then it is called a saddle point, or basically it means a transition point. That is, the system will not stay in that state for long. It will either go on to a maximum or a minimum. So this is how the three states are defined. And what the simulation program does is that it will continue searching. So I'll discuss that in the subsequent slides. So in this slide is basically, if you look at this energy profile diagram, so if you can see this gray line over here, on one side of the gray line is the reactant. This is called the saddle point or the transition point. And this is the product. So if you see, the reactant and product will both fall on either sides of a saddle point. And this saddle point is represented by taking the double derivative of the energy and it will be positive with respect to x and negative with respect to y, or vice versa. So the transition point can be is defined by that. And so we have seen this kind of diagrams, which is the potential energy versus reaction coordinate diagram. And this is basically taken from the three-dimensional energy profile diagram. From the three-dimensional energy profile diagram, it is reduced to two-dimensional by taking this particular gray line that you can see is marked here. So what is the procedure of energy minimization? Energy minimization is a numerical procedure to find the minimum on the potential energy surface, starting from a higher energy initial structure. So during energy minimization, the geometry is altered in a stepwise fashion till it reaches the global minimum. So suppose we are starting from point 1. The energy minimization proceeds by determining the energy and slope of the function at point 1. If the slope is positive, so you can see in point 1, the slope is positive. That means the coordinate is large and we need to go to lower values of the x-axis. So from 1, it will come to 2. In 2, the slope is still positive. So again, we need lower values of the x-axis, and it will go to 3, and it will continue this till it reaches 4. Now in 4, the slope is 0. That means the minimum has been reached. Now suppose our initial point was in the opposite direction, like our initial point was here. So the slope is then negative. That means the coordinate is too small. So we need to go to larger values of the x-axis. So then if we started from here, it would jump here, and then eventually it would reach the same minimum. So no matter whether we start from the right side of the graph or the left side of the graph, if we follow this numerical minimization technique, we will always reach a point where the slope is 0, and that is the minimum. All the energy minimization methods work on finding the minimum of this potential energy surface, and it continues this in an iterative way. So if the program stops as soon as it finds a minimum, then it can get stuck in a local minimum. So that is not what we want. So what the program does is that it will continue searching the whole surface till it reaches a point which is the lowest in energy, and there is no energy lower than that point. So that is how it finds the global minimum. All these are based on formulas of the type x new is x old plus correction. So if we consider the geometry as x, then the new x point is always the old x point plus a correction. Like suppose when the system moves from 1 to 2, the parameter of 2 is the parameter of 1 plus a certain correction. So the x coordinate keeps getting corrected till it reaches a value of energy which is the lowest. And like that, this is the procedure of energy minimization. So there are different kinds of energy minimization. There are first two kinds, called steepest descent and conjugate gradient minimization, are based on the first derivative. So these two methods calculate, so this is the general formula followed by these two methods, where it uses the first derivative of the function. So suppose you are starting from an initial point P0, which is defined by the x and y coordinates, x0, y0. So what this will do is that the derivative vector, this first derivative, will define the direction in which you have to move. So this is basically a contour diagram in which the middle part has the lowest energy, and as you move outwards into larger and larger circles, the energy becomes higher. So what happens is that the negative gradient of the potential energy points into the direction. So if you have P0 and P0 meaning B, yes. So what happens is that if you start from P0, and you take the first gradient, you will get the direction in which the function has to move in order to reach the minimum. So steepest descent, basically, if you look at this zigzag line, you can see a zigzag line moving from P1. So from P0, the function will predict to move to P1. Now P1 has lower energy than P0, but it is not the global minimum. So from P1, the function, as you can see, moves in a zigzag method till it reaches the global minimum, which is the center of the circle. The principle of steepest descent is that it uses the first derivative of the initial point to predict the final point. And then it will use the first derivative of P1 to predict P2, first derivative of P2 to predict P3. And like that, it will go on till it reaches the global minimum at the middle. The conjugate gradient minimization is similar, but it takes lower time. So if you look at this diagram, you are moving from R0 to the middle of the circle RST. The green line is the line followed using the steepest descent method. But the red line is the line followed using the conjugate gradient minimization. So it uses a similar principle, but it improves the step efficiency because fewer energy calculations and simulations are required to reach the minimum energy conformation. The disadvantage of these two methods is that it is based on the first derivative, so the calculation is much more rapid. In Newton-Raphson minimization, the advantage is that it is the fastest of all these minimization techniques, in that it hardly takes 1 or 2 steps to go from the initial to the minimized structure. But the problem is that the Newton-Raphson calculates the second derivative also. So it takes time. So because it has to calculate the second derivative of the energy, the Newton-Raphson method takes longer time, but it happens in like the number of steps is very much reduced compared to the other methods. So while it is computationally the most expensive step, meaning that a high processing computer is needed to get the second derivative, it also reaches the minimum energy function the fastest. So generally, in the energy minimization techniques of the simulations used in the papers you will see today, the Newton-Raphson method is the one that is used. So now we'll talk about the different steps in the molecular dynamics simulation. So in the first step, hydrogen is added. So if you have a PDB structure where hydrogen is missing in some of the atoms, so firstly the hydrogens are added. Then a box is defined. So if you can see, there is an outline of a box here. Defining the box is very important because we want to set a periodic boundary condition. We don't just want to let the simulation continue up to infinity, because that will take a huge amount of time. So a box has to be defined in which we will limit the molecule so that we can get the simulation result in a reasonable amount of time. Then the water or any other solvent, if that is what is needed, is added. And finally, the ions are added. So if we are thinking of replicating the system in our body, generally our body has many different salts like NaCl or KCl. So then those atoms will need to be added. Depending on what is the system we want to replicate, the type of atoms or solvent will change. So the principle of energy minimization is that you have the initial coordinate, which is prepared for simulation. This has high energy because, as I said, the initial model is not a very accurate model. It can have many interactions which are not favorable or steric repulsions. So the initial model generally has high energy. Then the energy minimization is performed to find the local minimum. So like we discussed over here, the slope is negative. That means the x coordinate has to move forward. So you move forward. then you find that there is a local minimum. To escape from the local minimum, simulated annealing has to be done. So we had learned about simulated annealing in previous lectures. So basically, the program will continue running till it finds it will run in random directions. So while going in random directions, it will find that when it comes to this position, it will find that this is of lower energy. So immediately it will go to the next local minimum. So like that, it will finally find the global minimum. And then when it tries running in random directions, it will see that this is indeed the global minimum and there is no other position that has lowest energy. So like this, it will be able to find the global minimum, and that is how it will reach the energy minimized structure. So in many cases, this energy minimization is done keeping the pressure constant. Like you can see here, the pressure is kept more or less constant, or the temperature constant. So that is called constant pressure energy minimization or constant temperature energy minimization. So there are many commands. So there is the software called GROMACS, which is generally used for the energy minimization. And there are several commands to carry out the steps that we had discussed in this slide. So like for adding the hydrogens, the command is PDB2GMX, or for adding the box around it, there is a command called editconf. So like this, you can try out there is a very nice free tutorial of GROMACS available. So GROMACS is also a free software. So you can try out the different commands and see that the different files that are generated. So I won't go into the details of the commands. I'll just talk about the results. So like we had talked here, the final structure that you have that is ready for the energy minimization will basically have solvent molecules, it will have ions, it will have hydrogens, and it will have a box, which is basically defining the periodic boundary conditions. So when you take that structure and then you carry out energy minimization, you will get a graph like this, which is basically the time is on the x-axis and the potential energy on the y-axis. So here 3 different proteins are shown. And for these proteins, you can see that the energy was initially high, but in kind of an exponential way, the energy decreases till it reaches a constant. So it takes around, say, 500 picoseconds for the energy to reach a minimum. And after that, the energy continues being constant. So it reaches the minimum energy around 500 picoseconds. Then the equilibration is done. So what equilibration basically means is that you are running the energy minimization in NVT conditions, meaning the number of atoms, the volume, and the temperature is constant. So if you see here, the temperature is more or less constant. It is hardly like 300 plus minus, not even 5, like plus minus 2 degrees, it is 2 Kelvin, it is varying. So throughout this time, the energy, the temperature is maintained as around 300 plus minus 2 Kelvin. So this equilibration is important because only when this is allowed, the system will be able to escape the local minimum and find the global minimum. So similarly, an NPT energy minimization is done where the pressure is maintained constant instead of the temperature. So here, if you see, it takes some time, like it takes around, say, 20 picoseconds to reach the constant pressure. And after that, it is more or less maintained constant throughout the time of the simulations. Again, this equilibration is important so that it can escape from the local minima and go to the global minima. So now that you have the energy minimized structure, the actual molecular dynamic simulation step is run. So that is done using an MD-run command. So if you see, this is a 100 nanosecond molecular dynamic simulation that is shown for this protein. So you can see how the different 4 residues are shown here: glycine, cysteine, histidine, and glutamic acid. You can see how the different residues are moving as from 0 nanosecond to 20, 40 up to 100. You can get different snapshots of the system at different times. And there are many different properties of the system that can be calculated from this molecular dynamics simulation, which we'll study about later. In the next few slides, we'll study about the RMSD and the RMSF, which are different properties that can be calculated from the molecular dynamics simulations. So now is the time to analyze the results obtained from the molecular dynamics and relate to the macroscopic level properties. Firstly, the trajectories need to be visualized. So VMD is the most commonly used software for molecular dynamics visualization. It provides many easy tools for trajectory analysis. So basically, to watch the GROMACS trajectory in VMD, the .gro file has to be selected. So we had gotten a .gro file at the end, and this has to be selected, and then the XTC or TRR file has to be loaded. So XTC or TRR is basically the trajectory files. So 1 second. Yes, you can see .gro is the PDB file that is energy minimized, and XTC and TRR are the trajectory files. So these together will give you the picture of the trajectory in VMD. So many times the picture of the trajectory needs to be processed further. So that is used, the command that is used is called trjconv, trijconv. So that is a post-processing tool, and what it does is that it strips out coordinates, corrects for periodicity, or manually alters the trajectory. So what does this mean? So many times the protein will diffuse through the unit cell and may appear broken or jump across to the other side of the box. So you can see here, the protein appears broken, like it is suddenly breaking off here and continuing on the left side. Again, it is breaking off here and continuing on the right side. So this picture needs to be manually corrected. So the periodic boundary conditions need to be adjusted such that the protein, these 4 images are kind of put together to get the actual image of the protein. And only after this can we study the trajectories. So this post-processing that is done, which converts a broken image like this into a picture of the protein, is done using the command called trijconv. So now we will come into the processing. So the final XTC file that is generated in the molecular dynamics simulation run, plus the TPR file, which has the topology, a GMX command is given, which is basically the command for analysis, and it produces an XVG file. So this XVG file is the one that is used for different kinds of analysis. So this is the processing step that is done. And after that, you will be able to calculate the different properties of the system, like the root mean square deviation. So root mean square deviation is basically defined by this formula, and it calculates the deviation between, say, the structure of the protein at 10 nanoseconds and the initial structure of the protein. So delta I is the distance between 2 different positions of atom I during the simulation. So suppose we take the initial position as our reference. So the difference of a particular atom at 10 nanoseconds from the initial position, at 20 nanoseconds from the initial position, all of them are added together and divided by the total time of the position, total time of the simulation, and whole square root is taken to get the root mean square deviation. So the root mean square deviation can be calculated from the XTC file using GMX RMS command to get a graph like this. So suppose you have two proteins, and in the y-axis, the root mean square deviation is plotted, and in the x-axis, time is plotted. So if you see for this red colored protein, the root mean square deviation is more or less constant. That means during the dynamic study of the protein, the structure is not changing that much from the initial structure. But in this black protein, there is a sudden jump in structure, and the root mean square deviation is initially small, but later it becomes large. That means there is a certain change in conformation of the protein. Like maybe the protein is changing from an alpha helical to a beta sheet protein. So that is why suddenly the root mean square deviation is becoming large. So this can be used to study which proteins are flexible. They will show a sudden change in RMSD, or which proteins are structurally very rigid, in which case the RMSD will more or less be constant throughout the simulations. Just give me 1 minute. The next macroscopic property is called root mean square fluctuation, or RMSF. So RMSF calculates the fluctuation of C alpha atoms in a residue of a protein in comparison with the respective average structure throughout the simulation. So increased residual RMSF value indicates the instability in protein backbone. So basically in RMSD, you are calculating with the initial structure, but in RMSF, you calculate with the average structure through the simulation. And higher the RMSF value indicates more unstable the protein backbone. So in RMSF is similar to the commands are similar to that of the RMSD, except in RMSD, the command was GMX RMS. And here it is GMX RMSF. So you will get a graph like this. And here you can clearly see that the black protein in this region, residue number say around 502 to 515 or so. In this residue, you can see that for this black protein, the protein backbone is very unstable. For red protein, it is comparatively stable, and it is most stable for the green protein. So you can find out from the RMSF, you can find out that which protein, again similar to the RMSD, you can find out that which portions of the protein are most flexible, and maybe those can be mutated according to what is the function that we need. Similarly, the B factor can also be found, like we had studied about the B factor, and that can also give an idea about how dynamic that portion of the protein is. Other properties can also be calculated, like hydrogen bonds. Since hydrogen bonds in protein structure are crucial in maintaining the functional 3D conformation of protein, as well as proper binding with the substrate, so we can study the change in hydrogen bonds. So with time, the change in hydrogen bond length can be studied. So we know hydrogen bonds mainly exist up to 3.5 angstrom. So any bond lengths that are above that are no longer hydrogen bonds. So our region of interest is the region below 3.5 angstrom. And over there, we can see that with time, which are the hydrogen bonds that are stable throughout the time, meaning that they are very crucial for the conformation of the protein, and which are the hydrogen bonds that are breaking. That means they are not that very important. So this variation in length of the intramolecular hydrogen bond or intermolecular hydrogen bond can be measured through the MD simulation run for a specific time scale. And using that, we can predict, like if it's a protein ligand interaction occurring through a hydrogen bond, and say this hydrogen bond is suddenly appearing after say 1000 nanosecond. That means the protein is binding only after that time period. So that is the time that we need to give the system to bind properly. So studying the hydrogen bond can give us all these information about the system. or it can be dihedral angle analysis. So we have studied about the Fisheye dihedral angles of a protein. Determining that for a specific residue is important to detect the mechanical importance of that residue to maintain the local conformation. So we have this glutamine residue, and three dihedral angles have been taken, chi1, chi2, and chi3, represented by three colors. So you can see the red and the black, which are the first two dihedral angles, are more or less constant throughout. That means in order to maintain the conformation of the protein, these two dihedral angles are very crucial. But the blue, which is the third dihedral angle, is changing with time. Like suddenly it is jumping to higher values, then it is jumping to lower values, meaning that this dihedral angle is very flexible. That is, this dihedral angle may be used for some function in the protein. So generally, the rigid dihedral angles are used to maintain the protein shape and conformation, and the flexible dihedral angles may be used in some function of the protein, like binding to the ligand. So using the MD simulation trajectory, this variation in the dihedral angle can be measured for the total MD run or for a specific time scale. Other properties can be studied, like converting a trajectory file to a PDB file. So to suppose you have a trajectory like this. So with time, how the RMSD is varying, you can see. So what can be done is that at each time, say at 25,000 picosecond, at 5,000 picosecond, a snapshot of that particular moment, like what is the orientation of the protein at that particular time, that snapshot can be taken and saved as a PDB. So that is also instead of studying, say, the constant time, if we want to see how the orientation of the protein is changing at different times, we can study the snapshots that are saved as the PDBs. The other thing is principal component analysis, or PCA. So PCA gives us idea about the protein backbone motion along the MD simulation trajectory. So to understand the pattern of protein folding, loop movements, etc. So what principal component basically means is that what are the degrees of freedom in which the protein molecule can move. So by studying these degrees of freedom, the motion of the protein molecule can be mapped. And so the, say, a loop is suddenly forming in order to bind with a ligand, then that can be tracked by tracking these degrees of freedom. So here is the plot of PC2 and PC1 with time. So states are representative conformation of a protein during a specific time scale in MD simulation. So say after a certain time scale, the protein is going from state 1 to 2 to 3 to 4. So if we want to find out in how to define these states, the basic concept is when these two degrees of freedom are crossing. So when the red and black lines cross each other, it goes from one state to another state. Then the second state will continue up to the time when they cross each other again. When the third state will start. Then the third state will continue, and when again they cross each other, the fourth state will start. So like that, the different states of the protein are defined. And within a particular state, the protein does not undergo any major conformational change, but it has slight motions within that particular conformation. So the change of state from 1 to 2 represents a major conformational change, but within 1 or within 2, the protein moves slightly but maintains the same conformation. So the free energy landscape can also be calculated from these molecular dynamic simulation. And it represents the equilibrium and dynamic properties of a protein in an accurate and clear way. These can be both 2D or 3D free energy landscapes, and it shows the stability and conformational changes of a protein with respect to Gibbs free energy. So if you see at the 2D state, So the blue color represents low energy. And as the color goes from blue to yellow to red, the energy becomes higher. So this is plotted with respect to the two principal components. And each of the blue spots, which are representative of either local or global minima, corresponds to a certain state of the protein. So we learned about the different states in the previous slide. So the states in the 2D free energy landscape are represented in terms of the blue contours, which are at the middle of this free energy landscapes. So if we get a snapshot of the PDB at this particular energy, we can see that they have different conformations and they are different states. If we want to get this picture in a 3 dimensions, we can also get a 3-dimensional free energy landscape. And these blue spots are now represented by these blue minima. And each of these minima correspond to a particular state, whether that be a local minima or a global minima. And in this 3D free energy landscape, the Gibbs free energy is basically a summation of KTB, where K is the equilibrium constant and TB is the gas constant, with a ln of PA minus PB, where PA is the probability of conformation A in the protein and PB is the probability of conformation B of the protein. So using the probabilities, you can calculate the Gibbs free energy at any particular point in this 3D free energy landscape. Next, we will look into normal mode analysis, or NMA. So this is a fast and simple method to calculate the large scale motions in biomolecules. So in this method, there is a function called correlation matrix, where basically what it will do is that it will represent the plot of correlation between motion of all C alpha in the protein structure. So suppose you have the initial protein structure and the protein structure after 100 nanoseconds. So that is superimposed and the correlation plot is drawn. So say residue 50 in the initial structure is corresponding with residue 50 in the structure after 100 nanoseconds. That means these residues are correlated, and so they are they are moving at the same time. So there is structural flexibility in the protein backbone. So if we look at these correlations, it represents different pairs of amino acids. So the same residue number is correlated on the X and Y axis. So this red line, it basically represents that residue 50 in the initial structure corresponds with residue 50 in the structure after the simulations. So residue 50 is not moving much. But if you look at these, so any residue which falls on this red line means that those residues are in the same position in the initial structure as well as in the structure after the simulations. But if you look at the spots around, like there is a blue spot here. So over here, a residue around 240 is corresponding with a residue around, say, 30. So these two residues are, the residue numbers are different, but they are correlating. That means these two residues in the course of the folding of the protein have somehow interchanged positions, meaning a major conformational change has occurred such that the initial position of residue say 240 is now occupied by residue 30. So that any points which are not on this line indicate residues that have undergone major conformational changes. Then there is a plot called the atomic displacement plot. Now we know that mesophilic proteins are the proteins that are present in normal organisms, and thermophilic proteins are present in proteins in organisms that can survive very high temperatures. So there are certain bacteria which can exist in, say, temperatures of even 100 degrees Celsius in hot springs. So they have thermophilic proteins, and the special property of thermophilic proteins is that they are resistant to very high temperatures, whereas mesophilic proteins get denatured at these high temperatures. So suppose you did not know that this was mesophilic and this was thermophilic. Looking at the atomic displacement plot, you can predict that, because what the atomic displacement plot shows is that how each of the residues are being displaced. So say you have, after your molecular dynamic simulation at the end of say 100 nanoseconds, what is the displacement of say residue 1, 2, 3 of that particular residue at the end of molecular dynamic simulation. So you can see that in mesophilic residues, the displacements are quite high, meaning that the protein structure is not that stable. But in thermophilic residues, the number of peaks is very much reduced. That means most of the residues have low displacement. That is, they are fixed at their positions. So the conformation is quite stable. So if you look at the atomic displacement plots, you can see that in the mesophilic proteins, the atomic displacement, mesophilic protein generally has, the number of peaks is generally quite high, meaning that the conformation is not that stable. But in the thermophilic protein plots, you can see that the number of peaks is very much reduced, meaning that the displacement is low. That is, the conformation is quite stable. So just looking at the plots, you can predict that which is the mesophilic and which is the thermophilic protein. Using this normal mode analysis, you can also study the movement of the loop. So suppose, so this is a protein in which there are two distinct conformations. One in which this loop is open and is ready for binding, and another in which the loop is closed. So how this loop is going from open to a partially open to closed, and again it slowly opens. So how this motion of the loop occurs during binding of the protein can also be studied using this normal mode analysis. So now we'll quickly talk about an application of this molecular dynamic simulation that has been used. So the target of the project was to develop cost-effective immobilized enzyme system which could capture carbon dioxide from power plant flue gas and prevent its emission to the atmosphere. So as you know, the concentration of carbon dioxide in the atmosphere is greatly increasing due to the industrial emissions. And so what was proposed is that if we use an enzyme to capture this carbon dioxide before it is released to the atmosphere, then the pollution levels will decrease. So the enzyme that was chosen for this is carbonic anhydrase. So carbonic anhydrase, what it can do is that it is in a family of zinc containing metalloenzyme, and it can stimulate the conversion of carbon dioxide and water into bicarbonate and proton. And bicarbonate and proton is water soluble. So if you take the industrial emission which contains the CO2 and pass it through carbonic anhydrase, then the bicarbonate and proton, which is water soluble, will be removed. So carbon dioxide will be removed in the form of bicarbonate using carbonic anhydrase. That was the proposal. Now, carbonic anhydrase are alpha-beta proteins with distinct folds. Most of the enzyme has beta sheets, as you can see shown by this yellow antiparallel beta sheet sign. The twist of the beta sheet creates an active site pocket at the bottom, of which lies the catalytic zinc. So it is shown by, the zinc is shown by this gray ball. And there are several long and flexible rules which loops in the structure which play important roles in the catalysis. So all these green loops that are shown are quite flexible. And as you know, a catalytic site always has to be flexible so that it can adjust to the shape of the substrate. And so these loops also play an important part in the catalysis. So in the active site of this carbonic anhydrase enzyme, zinc is coordinated by 3 conserved histidine residues. So you can see the 3 histidine shown here, which are coordinated to the zinc, and one catalytic water molecule. The role of the zinc is as an electrophilic catalyst. So we look at the reaction mechanism of carbonic anhydrase. In carbonic anhydrase, what happens is that, so this is the initial structure of the enzyme where Zn2 plus is coordinated to 3 zinc and 1 water, as shown here. So initially a deprotonation occurs and the H plus leaves, and so this becomes O minus. Then the CO2 binding happens. So this O minus attacks the CO2 molecule, and CO2 becomes COO minus and is coordinated to the zinc. After that, this entire group, HCOO minus bicarbonate, this entire group leaves. So what happens? This entire group, HCO3 minus, leaves and a water molecule enters. So water molecule enters and coordinates to the zinc, and this entire group leaves. So water enters and bicarbonate leaves, and the enzyme is again regenerated. And so it can again convert one molecule of CO2 into HCO3 minus. So that is why, since the enzyme is continuously being regenerated, only a catalytic amount of the enzyme is sufficient for the catalysis. So this is the mechanism by which the carbonic anhydrase is converting the CO2 to the HCO3 minus. So the focus of the current study is to look for a carbonic anhydrase enzyme which is stable thermally and with pH, so that it can be used industrially to reduce the excessive CO2 emission. To achieve that goal, in silico screening process is required to understand the structure stability. So there are many different carbonic anhydrases from many different organisms, but you need a particular carbonic anhydrase that has both stable structure with respect to heat and pH, and also the efficiency of conversion of CO2 to HCO3 minus has to be good. That is, the turnover number of the enzyme has to be high. So in order to understand and screen this stability of the carbonic anhydrase, molecular dynamic simulation is performed. So in this particular example, two carbonic anhydrases have been shown. One is from Neisseria gonorrhea, Ng alpha Ca, which has high activity. That means it can convert, like the output is very high. So if a certain number of CO2 molecules are taken, it will convert most of them to the HCO3 minus. And the other is from sulfurihydrogenibium species, or SSP alpha Ca, that has high thermostability. So that means it is very stable under high temperature. So you actually need both these properties in a certain in one enzyme. So first, the MD simulation technique was carried out at different temperatures, 300, 350, 400, and 500 Kelvin. And it was carried out for 100 nanoseconds. And the GROMACS software was used, and the water was used as the solvent. And we have learned about the steepest descent algorithm. So steepest descent algorithm was used. So if you look at it, then you can see that at 300 Kelvin, the RMSD is not much for either protein. But as the temperature is increasing, this black protein, which is the Ng species, has higher and higher RMSD, which means it has lower thermostability. So what is happening in the thermostable SSP species, the RMSD is remaining comparatively lowered even at high temperatures. So as we learned, RMSD is the deviation of the conformation from the initial structure. So as the temperature is increasing, the final structure is unfolding from the initial structure in case of the Ng species, which is mesophylic and has less thermostability. And for the higher thermostability, the final structure is comparatively closer to the initial structure, and it is more thermostable. Similarly, if RMSF is compared, you can see that for the Ng species, as the temperature is increasing, the protein is greatly fluctuating, meaning that the backbone is becoming more and more unstable. But for the SSP species, which is thermostable, at high temperatures, the backbone is comparatively more stable. Salt bridges were also detected in the two different structures. So we have learned that hydrogen bonds can be detected through molecular dynamic simulation. Salt bridge is a kind of hydrogen bond, so salt bridges can be detected. So for the NG species, it was observed that salt bridges between arginine and aspartic acid. So you know arginine is a basic residue and aspartic acid is an acidic residue. So 4 salt bridges were observed with different distances. And for the SSP species, 5 salt bridges were observed between different acidic and basic residues like lysine with glutamic acid or arginine with glutamic acid of different distances. So what this dynamic study is providing you? Such simulation is helping us understand the difference of atomic movement between the mesophilic and thermophilic carbonic anhydrases. It is also giving information about important non-covalent interactions like salt bridges and are showing their effect in a time-dependent manner. So now what we need to do is we need a carbonic anhydrase which has high activity like Ng, but which has the thermostable property of SSP. So mutation needs to be done so that Ng has to be mutated in such a way that the thermostable property of SSP is incorporated into Ng, so that we have both high activity and high thermostability. So 3 mutations were done in Ng to make it similar to that of SSP. The serine at 44 was mutated to arginine, the lysine at 168 was mutated to arginine, and the serine at 139 was mutated to glutamine. What this happened is that if you look at the original mesophilic Ng, it had a melting temperature at 312 Kelvin. That means beyond 312 Kelvin, the protein is completely unfolded. But in the mutant mesophilic Ng, where the residues have been mutated so that they become more like SSP, the melting temperature increases from 312 to 333. That means it is stable up to 333 Kelvin and loses its conformation after that. So a mutant has been created which will have the high activity of Ng, but a thermostability has also been introduced so that it is more stable up to a higher temperature. So using molecular dynamic simulation, this mutation could be predicted that at what points the mutations would be. So if you look at the RMSD and RMSF, it gave an idea about which are the flexible residues which can be mutated so that this thermostability property can be introduced. So that was the application that helped design a carbonic anhydrase, which had the property of both high activity as well as high thermostability in order to make an efficient carbonic anhydrase to absorb CO2 from the industrial emissions. And to talk very briefly, nowadays, the simulations that are done is a hybrid approach. So basically, it's guided by the general idea that large chemical systems are partitioned into an electrically important region, which requires a quantum chemical treatment, and a residual region, which requires a classical chemical treatment. So nowadays, so we have mostly talked about the classical treatment so far. We haven't gone into quantum chemical treatment. So nowadays, like we talked about the Newton's laws of motion. So that is a classical physics treatment. But as the molecular dynamics procedures are improving more and more, nowadays a hybrid approach is used in order to more accurately predict the dynamics of the system. And this hybrid approach includes both classical as well as quantum mechanical treatment. So I have talked about like a summary of week 10. So now we'll just look into the sample questions. So the first question is that molecular dynamic simulations are based on which of these four? So the answer here is B, or Newton's equation. So like I discussed in the previous slide, the hybrid approach, which includes quantum mechanics, is called a hybrid QM-MM approach. Molecular dynamics simulation, which is what we studied, is based only on Newton's equation. So just to recap what we had studied about the Newton's equation. So for a atom I, the force on the atom is a product of mass into acceleration. And acceleration can be written as the double derivative of the position r at time t. The force can also be written as the negative gradient of the potential energy. And so combining equations 2 and 3, you can write that the negative gradient of potential energy is mass into the double derivative of position r at time t. So using this, you can find out the position and momentum of each of the particles I at a time t. So these are the Newton's equations of motion that are used for the molecular dynamics simulation. And so the correct answer is B. The next question is, when we convert the PDB file to .gro file, which file will not generate? So the correct answer is B. So if we look into the commands that are used to convert the PDB to the .gro file, we can see that the final file is .gro, but we also get the topol.top file, which includes the topology of the atoms. So like the different atoms that are connected by bonds. So the topol.top file is generated, and the posray.itp file is also generated, which contains information about positional restraints. So if a certain position is fixed by a hydrogen bond or by some other covalent interaction, then that positional restraint is mentioned in the posray.itp file. So in this set of commands used to convert the PDB file to the .gro file, you indeed get a .gro file, you get a topol.top file, and you get a posray.itp file. But you don't get an XTC file because that is obtained after the molecular dynamics simulation. So the correct answer is D, which is .xtc. The next question is that what RESNR defines in the topol.top file. So the correct answer is B, which is amino acid residue number. So this is a topl.top file. So you can see RESNR written here, and the first residue is glycine. So residue number is 1 for all the atoms of the glycine that is being discussed. So RESNR discusses the amino acid residue number. So the correct answer is B. Now phase space is a how many dimensional space? The correct answer is D, 6-dimensional space. So phase space will basically, suppose there are two particles, P1 and P2. Each particle will have three parameters: the position P, the momentum Q, and the time. So since it has three parameters and two particles, it will be a 6-dimensional space. Which physical property is constant in microcanonical and canonical ensembles? The correct answer is A, which is number of atoms and volume. So if we look at the microcanonical and canonical ensemble, it is both are sealed, and so the number of atoms and volume is constant. But in microcanonical, there is an insulation, and so there is no energy exchange with the surroundings. So the energy is constant. But in canonical, there is no insulation, so there is energy exchange with the surroundings. And so energy is not constant, but temperature is always constant, because it can exchange, interchange that energy with the surroundings till the constant temperature is reached. So if it is asked that which physical property is constant in both, then it is N and V, which is the number of atoms and volume. So the correct answer is A. The next question is, what are the conditions for minimum, maximum, and saddle stationary points in potential energy space respectively? So the correct answer is B. So if we recap what we learned, is that for the minimum, the double derivative of energy with respect to both x and y has to be greater than 0 or positive. For maximum, both has to be less than 0 or negative. For the saddle point, one has to be greater than 0 and one has to be less than 0. So if we look at the option B is the only one where for minimum, greater than 0, greater than 0. Maximum, less than 0, less than 0. And saddle point, one is greater than 0 and one is less than 0. So the correct answer is B. Question 7 asks, which of the following is known as bonding energy term? So the correct answer is C, or angle bending. So if you remember, the three bonding energy terms are stretching, bond stretching, angle bending, or rotation along a bond, which is the torsional distortion. So in these options, the only one available is angle bending, and so the correct answer is C. Question 8 asks, which of the following is not implicit solvent model? So the correct answer is C. And if you recap, explicit solvent model is when each of the solvent models are defined as different molecules. And in implicit solvent model, it is defined as a continuum around the protein molecule. So the different kinds of explicit solvent models are fixed charge models and polarizable water model. And the implicit solvent models are Poisson-Boltzmann general bond model, Karplus model, and Benoit-Roux spherical solvent boundary potential. So you just have to remember the names. And if you look at the question, which of the following is not implicit, meaning which is explicit? So the correct answer is polarizable water molecule, which falls under the explicit solvent models. So you just have to remember the names under the explicit and implicit solvent models. Question 9 asks, the topology file may specify what? So the correct answer is A. So actually it can specify bond, angle, and dihedral all. But these definitions are wrong. Like dihedral is not 3 atoms, it is 4 atoms. And angle is not 4 or 5 atoms, it is 3 atoms. So option B, C, and D are wrong. So option A is correct, that the topology files specify bonds, which are the 2 atoms connected. And question 10 asks, what is the post-processing tool for the analysis? So after molecular dynamic simulation, what is the tool used for post-processing? So the correct answer is A, TRIGConf. So as we discussed, what TRIGConf does is that if the protein is breaking like this, that is the trajectory abruptly ends here and continues on the left side, or abruptly end on the left side and continues on the right side. What TRIGConf does after processing is that it puts all these images together to get a final complete image of the protein trajectories. So TRIGConf is the post-processing tool, and so the correct answer is A. So that ends today's sessions. If you have any questions, you can always write an email to me, or if you have any doubts, and I will clear them in the next session.